All right. So, finally, part 3 of 3 for the chapter 2 stuff. So, again, all of chapter 2 has to do with cells. We had a little intro stuff going on. We had talking about prokaryotic cells, and then now is the chapter, or the section of the chapter about eukaryotic cells. So, just to jump right in, like we've said before, eukaryotic cells are more complex than prokaryotic cells. They tend to be a lot larger, like orders of magnitude larger. Um, they contain membrane-bound organelles, okay, which are little machines designed for a specific purpose. Um, a lot of times, they use vesicles to kind of shuttle things around. So if you remember, a vesicle is basically just a piece of a membrane that it gets broken off of something and it encircles whatever is being moved, and then it joins the membrane of this over here. And so that little pathway between it was called a vesicle. So the actual definition is a sac surrounded by a membrane used to transport substances. There are some things in eukaryotic cells that do not have a membrane, such as ribosomes, which are present in both prokaryotic and eukaryotic cells, different filaments that make up the cytoskeleton, that sort of thing. But the thing that really sets eukaryotic cells apart is that they do have membrane-bound organelles. Okay? So to start off with, I feel like we've talked about this ad nauseum at this point, but the plasma membrane, just to review, okay, it's a phospholipid bilayer. It separates the inside of the cell from the outside of the cell. It contains integral proteins that go, here's our layer. Integral protein goes all the way through. A peripheral protein sticks out on the top. Okay, so integral proteins are channels, carriers, transport things. Peripheral proteins can be receptors or identification things. Eukaryotic cells, just like prokaryotic cells, they can form a glycocalyx. So they put a coating of carbohydrates on their surface. That is really important to that cell kind of identifying itself. So telling our immune system, don't attack me, here are my credentials. With my glycocalyx, you can recognize me. A hugely important example of the glycocalyx kind of identifying cells is our AB blood types. So certain red blood cells have a marker on their surface made up of carbohydrates that we designate A. Some have B. Some have both A, B. Some have neither O, which is obviously hugely important to giving and receiving blood. Okay. In a lot of tissues, especially in animals, okay, so cells come together to form tissues. So we have a lot of cells all together. There tend to be cells called fibroblasts that they secrete the goo in between cells. And so it's not really a great place to put it in this talk, but it's the only place I can think of. So the goo in between cells secreted by the fibroblasts is called the extracellular matrix, or the ECM. It's made up of kind of inorganic fibers and things that kind of hold everything together. Okay. So kind of our first big membrane-bound organelle to talk about is the endoplasmic reticulum, or the ER. Basically, it is just a system of connected membranes, and various portions do different things. The inner space kind of inside the ER membrane is called the lumen. So just kind of side note, you will see this term come up a lot, a lumen. So that really just means the inside of a tube or a membrane or something. So for drawing blood, we need a needle to go into the lumen of the blood vessel. The lumen is where the blood actually is. It's not in any of the layers of the vessel itself. So the lumen is the inside. So side note about that. There are two major types of endoplasmic reticulum, rough and smooth. So rough endoplasmic reticulum, rough ER, it's designated that because when you look at it, it looks like it's got little speckles all over it. those little speckles are ribosomes. So ribosomes are what cells use to make proteins. So that portion of the endoplasmic reticulum, important in making proteins. Okay? That portion of the endoplasmic reticulum, also has other things to kind of add to the proteins, adding carbohydrates, if it's glycoprotein, adding other stuff. That might be where you find some chaperone proteins that help ensure the protein fold correctly into the shape that it needs to be. Okay. A huge portion, we've said this already, a huge point to be made is that form equals function. So how a protein is shaped determines what it does. And our body is very particular about proteins that are not shaped correctly. Okay? So in the endoplasmic reticulum, they have chaperone proteins to kind of help fold things. There's also things there that kind of scan and look at these proteins and they see if anything's wrong with them. So if there's something wrong with it, they can target, send a chaperone protein and maybe refold it. Or if it's too far gone, send something like ubiquitin to target that protein for destruction. If there's just too much damage, tons of misfolded proteins, the endoplasmic reticulum can actually signal and say, done all I can, apoptosis time, which is programmed cell death. So the cells would actually rather be dead than to have inappropriate function. Okay, that's kind of what sets regular cells apart from cancer cells. Cancer cells say, we're going to survive and reproduce and have irregular function no matter what. So a lot of that has to do with the rough endoplasmic reticulum, as well as the smooth endoplasmic reticulum. It looks smooth because it doesn't have the ribosomes on it. So it's not hugely important in protein synthesis. It's more important in producing other things, lipids, that sort of thing. If a cell is a cell that needs a lot of proteins, its rough ER will be a lot bigger. If it's a cell that needs a lot of fats, its smooth ER will be a lot bigger. So it just kind of depends on what the cell is to what its components are. Okay? The smooth endoplasmic reticulum in muscle cells is actually very, very specialized. So, like I said, if you need a lot of proteins, you need a lot of rough ER, and so on. The smooth endoplasmic reticulum in skeletal muscle cells, so just regular muscle cells, it's so specific and unique to that cell, it actually has its own name called the sarcoplasmic reticulum. So it stores calcium, which is important for muscle contraction. We'll get to all that later. All right. So the next organelle, it's kind of a continuation of the ER, is the Golgi apparatus. So in my brain, the Golgi apparatus is like the Amazon warehouse storage depot of the cell. So it takes everything that the endoplasmic reticulum made or other organelles made or whatever, packages them and kind of sorts and sends them where they're supposed to go. So, like Amazon. All right. If the substance needs to go somewhere else in the cell, it puts it in a vesicle and ships it to that other organelle or wherever it needs to go. If it's something that it needs to ship out of the cell entirely, it's able to do that as well. So that kind of leads us to a separate point about things going completely out of the cell or things coming into the cell. So the plasma membrane can let little tiny particles filter through, but what if the cell needs something big in or out? Okay, well that's where endocytosis and exocytosis come into effect. Okay? So endocytosis is when something large needs to come into the cell. The cell kind of moves its plasma membrane around it and kind of pinches it off, and that becomes a vesicle, and then that vesicle travels to wherever it needs to go. Okay. A type of endocytosis, if it's bringing in a liquid, is technically called pinocytosis, but the one that we need to know is called phagocytosis. This is when a white blood cell engulfs a bacteria or a damaged cell. So our immune system engulfing debris that shouldn't be there is called phagocytosis. Okay, the opposite of that, so if the Golgi has packaged some hormones that need to be distributed to the rest of the body from this cell, then it does that via exocytosis, where the organelle has all the stuff inside of it, It travels to the plasma membrane, it fuses with the plasma membrane, and then it releases those particles, hormones, whatever it needs to, into the bloodstream. Okay. So you can kind of see that the plasma membrane and all the other membranes are always kind of in flux. So a bit of this one gets taken off and added to this, and then a bit of this gets taken off and added to that. So the plasma membrane is constantly being remodeled, is what they say. All right. Lastly, there is another kind of endocytosis. I shouldn't have mentioned it, but it's called receptor-mediated endocytosis, and that is where our ligand binds to the receptor, and then the receptor triggers response to then engulf, that receptor and that ligand and whatever it was. So again, the clue's in the name. Receptor-mediated endocytosis is just endocytosis that's triggered when a receptor is activated. Moving on, there are some kind of debris cleaning up organelles. So there are things called lysosomes. So basically, they're a vesicle that contain digestive enzymes, and those digestive enzymes break things apart. So if there's something that the cell needs destroyed, put it in a lysosome, let those enzymes kind of break it apart. In plant cells, they have a large lysosome, maybe called a vacuole, where it's also a water storage depot, and it lets the cell kind of maintain its shape and rigidity. It's called turgor. In addition to lysosomes, there are also something called peroxisomes, which are a vesicle that contain enzymes to get rid of debris, a different type of debris, so different types of enzymes. Peroxisomes are what break up, like hydrogen peroxide, free radicals, those things that can really damage the cell. So it's very important that the things in the lysosomes and the peroxisomes stay in them, because we don't want those enzymes just kind of running free and destroying everything in the cell. Okay. My favorite organelle, is the mitochondria. You have seen the memes of I don't know anything else, but I do know that the mitochondria is the powerhouse of the cell. Okay. So I put it in bold, all caps on the notes. So the mitochondria is the powerhouse of the cell. So what does that mean? Well, the mitochondria is the organelle where ATP is made. So what is ATP? Well, we'll talk about that ad nauseum. But ATP is the fuel that, or is the energy source that cells use. Okay. All of the food that we take in gets processed and waste gotten rid of, and then we break down all these molecules. We have all these chemical processes, and the point of it is to use those as fuels to make ATP. And then our body's able to use ATP to do whatever it needs to do, at the cellular level. Okay, so mitochondria is where this process actually happens. Okay, so the mitochondria is kind of unique. It has two membranes. So if you think of a mitochondria, it looks like a little bacteria, like a little rod-shaped bacteria inside the cell. So it has an outer membrane, and it has an inner membrane that's in little squiggles. Pictures on the internet do a lot better justice than my hand waving, but that's just kind of the basic layout. Okay? The outer membrane is kind of unremarkable. Okay? The inner membrane, those little squiggles are called cristae. All right? And now, inside the inner membrane, but outside the cristae, is called the intermembrane space. And then inside the cristae themselves, so inside that inner membrane, is called the mitochondrial matrix. Okay? The inner membrane itself, so the actual squiggles of the membrane, they contain embedded in them the integral proteins and the peripheral proteins that are embedded in that membrane are the machinery that work like ATP synthase to actually make ATP. And so we'll talk a lot about what's in the matrix and what's in the intermembrane space and how they come across and how that generates ATP. And that's the whole rest of biochem. Okay? So mitochondria actually have their own DNA, so they kind of have their own instructions. I thought it was interesting because you inherit all of your mitochondria and therefore all of your mitochondrial DNA from your mom. So you technically have a little bit more DNA from mom than you do from dad. So be nice to mom and dad and say, Mom, I'm technically a little bit more related to you than I am to dad. Because mitochondria have their own DNA and it comes directly from mom. All right. Mitochondria If it's a cell that requires a lot of energy, there's a ton of mitochondria. If it's a cell that doesn't require much energy, not so much mitochondria. Okay, pretty self-explanatory. Our body is very good about not being wasteful and keeping things around that it doesn't need. All right. Just as a side note, I'll put this in here. There's an organelle group called plastids. Plastids allow photosynthesis. So in plants, that organelle is called a chloroplast. In different amoeba or whatever, plastids that can do photosynthesis is called something else. The main one that we need to know is the chloroplast. We'll talk more about it when we talk about photosynthesis. But basically, the chloroplast is an organelle that our cells do not have, so we cannot perform photosynthesis. Plant cells do, so it contains things like thylakoids, terms like grana and stroma. So again, more on that when we talk about photosynthesis. We'll talk about the plant cell and the chloroplast itself. Okay. On that note, plant cells, yes, they have a chloroplast, so they're a little bit different. Plant cells also have a cell wall. Okay. It's not a peptidoglycan cell wall like the bacteria, but it is a cell wall nonetheless. Okay. So even though they're eukaryotic, plant cells do have a cell wall. Right. So just kind of moving on, there are little bits in the cytoplasm of a eukaryotic cell called the cytoskeleton that are kind of like latticework structures, kind of rebar and steel beams and wooden planks and everything that is supposed to hold the cell together. Okay, so you can really get into the weeds about, okay, there's microtubules and what they're made up of, about what their subunits are. There are microfilaments, which are different, and what their subunits are. There's intermediate filaments. All I really care about is us knowing, okay, we have these filaments, these things that kind of hold the cell together. Okay? Their jobs really are to maintain cell shape, to allow for cell movement. So if the cell was kind of, needing to engulf something, it's got to move that plasma membrane. Well, the cytoskeleton elements are kind of poking the membrane out and allowing it to move around and that sort of thing. Okay. The cytoskeleton is also there to provide a framework for other things to happen. So if you watched the hilarious molecular machines video, you saw the little protein that was walking on the little tightrope and dragging the huge thing behind it. The little tightrope would be like a cytoskeleton element. Okay? So that's all that we really need to know for the cytoskeleton. All right? Next to last are the ribosomes. So the ribosomes also present in prokaryotic cells. Ribosomes, their job is to make proteins. So in the rough ER, there's lots of ribosomes, make proteins. There's also ribosomes scattered throughout the cytoplasm. What's their job? Make proteins. Okay? Ribosomes have their own kind of genetic material called ribosomal RNA. So a ribosome is actually ribosomal RNA and proteins, and they're made up of subunits, and these subunits all come together to form the complete ribosome. What actually happens is DNA from the nucleus is a portion of it is transcribed into RNA. RNA comes out to the ribosome reads that RNA in a process called translation, and the ribosome actually puts the protein together. It takes, okay, I need this amino acid first, I need to attach this amino acid second, this amino acid third, and it makes the protein that way. Okay? So we'll talk a lot about transcription and translation kind of moving forward. And then lastly, maybe most importantly, is the nucleus. So prokaryotic cells, their DNA is kind of out in the cytoplasm. It's a little circle. Okay? Eukaryotic cells, we have a lot more DNA, and it's housed in a nice special container inside the cell called the nucleus. Okay? The membrane of the nucleus is actually called the nuclear envelope, and it has little pinprick holes in it that are called nuclear pores. Okay? The DNA never actually leaves the nucleus. Okay? If genes or genetic material needs to be out of the nucleus, then an RNA is made, and the RNA leaves the nucleus and then goes to the ribosome or whatever it needs to do. Okay? The only time that there isn't a nucleus for most eukaryotic cells is during mitosis, when the nucleus kind of, that nuclear envelope degrades. We have double the genetic material and then 2 nuclei form around it. Okay? But also, we'll talk more about that later. All right. Inside the nucleus, so in my brain, I think, okay, that's where the DNA is. That's where we see the chromosomes. Inside the nucleus, you just see kind of a goop, kind of a nasty-looking mixture of proteins and genetic material. That mixture is called chromatin. Okay. Technically, chromatin is the DNA mixed with proteins such as histones. Okay. It's not until mitosis when that chromatin kind of comes together and then forms the nice chromosome shapes that we're familiar with. Okay. The rest of the time, the genetic material is just kind of blob form hanging out as chromatin. There is kind of a subsection inside the nucleus called the nucleolus, is where the ribosomes are actually put together. Okay? So I think that is it for eukaryotic cells. Fortunately, it didn't take too awful long because we covered a lot of this beforehand, and we should know most of this. So sorry if this was mostly just a refresher. So moving forward, our next chapter is about water, and then I think we'll lump all those things together, chapters 1, 2, and 3, and then kind of do a review from there, and then hopefully we can launch into some more heavy biochem stuff, and hopefully we can get a classroom soon. All right. Okay, so I will post this and hear from y'all shortly. Bye.